Again, I'd like to welcome everyone here. If you're here for the first time, great to see you. If you've come back, welcome back. I'm astonished to say is the fifth of these 2 weekly alumni meetings called Coffee with EMBL, and I have my EMBL mug and my coffee with me again, and it's been a great pleasure for me to help to host these events. I found them really exciting, and the discussions really interesting and productive. And once again, we have a fantastic lineup of speakers and some great topics for discussion. I see that there's a lot of people just still joining in, but just to say again that this is now the fifth in a row of these series of meetings where we've been discussing COVID-19 and its impact on the world community from the perspective of alumni across 5 different continents and more than 30 different countries. I think it's been remarkable, the ability to have this dialogue via the online video conferencing platform. And just for those of you who haven't been present at all the other meetings, just to remind everyone, we've covered an amazing range of topics. We've had a fantastic overview of the biology of coronaviruses. We've heard from alumni from all the way from the epicenter of the outbreak in Wuhan in China across to Australia, where there are alumni there. Keith Stanley was working in his company, making test and diagnostic kits to help in the fight against the virus and tracking the virus. We've heard from alumni working in a number of different countries, including in Italy. We're working on vaccine development, and we're going to come back to the theme of vaccine development later on today. And we've also heard a lot about data analysis and interpreting data, about how we interact as scientists with the media and communication of science. So really, it's been quite remarkable, the range of topics that have been covered. And we have also had the opportunity to hear from leading researchers in senior positions at all of the major EMBL laboratories, and we'll conclude that tour around the EMBL laboratories today, talking with Matthias Dolmans from Hamburg, and he'll be speaking very shortly. So just to let you know again what's planned going forward, this will be the last of this current series of sessions in this format, focused on the COVID crisis. In 2 weeks' time, on the 3rd of July, by a happy coincidence, EMBL has organized an international conference on the topic of COVID-19. And I'm going to introduce in just a few moments Nasus Tipas, from a group leader at EMBL and one of the co-organizers of this conference, to tell you about it if you haven't heard about it yet. I'm also very pleased to say that they have generously agreed to keep the registration for this conference open until next Monday. It's limited to 1,000 places. There are some places still available. And so it's great that for anyone who hadn't heard about it and would be interested to join, on Friday in 2 weeks' time, instead of this meeting, you can potentially join that. And Nasus will tell you about that conference, which is a great, I think, endpoint and what otherwise would have been the 6th of the meetings that we've had here focused on the COVID-19 crisis. 2 weeks after that, on Friday, the 17th of July, I believe it is, that will also coincide with EMBL World Alumni Day. And so this Coffee with EMBL session will be replaced by the EMBL World Alumni Day, which has been held in the last several years as an event actually on campus. It includes the award ceremony where alumni are presented with prestigious medals who have been selected for those medals. We'll also hear from the Director General, Edith Herd, about the plans for the future of EMBL. And we'll have a World Alumni Hour. So there'll be more to say about that, and I'm sure Noosh will be in contact with you to update you on all the plans there are for that day. Is there anything you would like to share at the moment, Noosh, about World Alumni Day?
Just to also say hi and welcome everyone. It's wonderful to see so many people from so many countries and continents, and quite frustrating that I can't go up to you and talk to you all. And to let you know that the alumni team is here. You will have dealt with them in various ways. And there's Peter, who's going to move a hand if you're all on gallery view. And there's Fenya, who's going to wave at you, and Tom, who's going to wave at you. And also there's various members of the alumni board here. Hopefully they will join in with the chat and bringing up questions. So welcome on board. And Angus, you're covering everything beautifully. Thank you so much for hosting and for moderating this.
You're very welcome. So again, in the interest of saving time for some of the discussions, I hope that we'll be able to have. Again, I encourage people, if you're not familiar with it, to open also the chat dialogue box and use that to communicate with either to message privately to people you see who are participating, or to post a message for the speakers. We will try to have some questions for the individual speakers and make this as participatory as we possibly can. And just as we've done in the last couple of meetings, as well as the main hour of discussion, we'll have the EMBL second cup of coffee at the end of the hour. And that will also, we hope, be a chance to have more general discussion. And I'd really like to give an opportunity there for anyone participating who'd like to speak or ask questions. And we encourage the speakers, if they possibly can, to stay around for that and be able to answer more questions there. Fantastic. So without any further ado, I'd like to move on to ask Nasos Tipas to tell us about the EMBL COVID-19 conference coming up in 2 weeks' time. Nasos.
It's great to be here. So I'm Nasos Tipas. I'm a group leader here at EMBL in genome biology. So I'm working peripherally with infectious diseases, and as I think the whole pandemic hit, we're discussing about the new program at EMBL and some of the new directions that EMBL could go, and part of that was infection research and infection biology. So there's been discussions going on whether we could set up a conference to try to bring a lot of the cutting-edge research that is done nowadays on COVID-2, but also use it as a stage for discussion about the future of infection research at EMBL. And this is what got the conference going. So I've sent to everybody in the Zoom chat a link for the conference. You can go and see there I think we have a very good speaker lineup. So it's organized by another colleague of mine at EMBL, co-organized, so Pedro Belchar from EBI, then it's Pascal Cossart from Pasteur Institute, and Dirk Heinz from the Helmholtz Institute for Infection Research. If you will see, the program has different angles, so we try to cover quite broad. So I think it's quite centered around the new pandemic, and it goes on quite a bit on the molecular and cellular biology of the virus, going towards fundamental research that has to do with trying to find drugs and vaccines, to epidemiology and surveillance. And I think then it opens up, because this is not the first infectious disease agent, not the last. So there are going to be more. So the discussion is a little bit is what we've learned from the past, what we're learning now through the pandemic, and try to use that information to discuss what are our current limitations and where infection research is heading in the future. So in the last session is more open up. It has to do with people working on bacterial, fungal, or parasitic pathogens. We discuss about AMR and microbial resistance and how this may be affected also by the current pandemic. And then at the end, we have a round table where we have a few different topics that we want to discuss that, again, open up a little bit the discussion. So we want to discuss how science is published nowadays in the times of pandemics, how we disseminate that information to public. I think there were a couple of incidents in the last months that sparked quite a bit of discussion. And of course, science opinions matter nowadays. They make it down to policy decisions. So we want to discuss also about that part. And I think what is becoming apparent is that we have limitations from the beginning of the pandemic on things that we're not expecting. So how we can coordinate efforts where different disciplines can help. So, for example, in detection of the viruses, whether we do have in academia approaches and methods that can move much faster to that detection. And then also discuss about what is the roles of pharma and academia and trying to interface and collaborate for infectious diseases. So this is a little bit about the kind of ideas we had about for the meeting. There are quite a few prominent scientists talking. We try to bring virologists, but not only virologists. So there's people coming from many different fields doing actively research now on COVID-2, but also coming from outside, trying to bring technologies or approaches that would help a lot on that direction. So and on the round table, there's also quite a few people that have to do also with policies, so we can discuss a little bit further. And so with that, I mean, I would be happy to answer any questions. What I want to say is, Agnes already mentioned that, and Nush, so the conference is open, and I thought there was a link, so that I put there, and so the registrations are open, and they're open till Monday, and there's still a few slots basically to fill out. And we'll be happy to see you there.
That's fantastic, Nessos. I have not only registered, I've even paid my registration fee. So that I can't do on that. So I hope it will also be something positive for you to relate back to your colleagues at EMBL, that the alumni of the EMBL have also been very active at multiple levels, as we've been hearing about in this series that's been taking place over the last 2 months, as I say, on a whole range of topics, from actually carrying out basic research into the virus to working in companies developing vaccines, antivirals, test kits, and detection kits. So yeah, I think the EMBL community at all levels has played a big role in this, and I think that issue of communication of science is also really critical, as how we interface with our political masters who ultimately make policy and fund things. So really looking forward to it. Again, if anyone has questions, please let us know, and we'll try to get back to that. So moving on from NASA's, then we throughout these series, we've had senior members of each of the main EMBL laboratories. We've been around talking with people in Barcelona, in Rome, in Hengsten at the EBI, in Grenoble, and of course in Heidelberg. And today it's a great pleasure then to welcome Matthias Wilmans from Hamburg, who will come now and tell us about how life is going in Hamburg and some of the work going on at EMBL Hamburg, also to contribute to this topic of COVID-19 research. Matthias, hello.
Okay, so hello all together, and thanks for inviting me here, for the coffee with the alumni, and it's really, I'm super happy. Now we have 77 participants. I've been scrolling down. Hi, Santosh from Australia. Can you? So okay. And it's really great, to see, names, with, I personally connect, one or the other thing. And, okay, so what I've got actually 10 minutes, and I've organized a small presentation, which I just have to share. Just give me a second. So, okay, what I'm going to do in the next few minutes, to those of you who maybe have heard about Hamburg, never heard about Hamburg, or had only heard about Hamburg many years ago, I just give a very short introduction about what we're doing in Hamburg, but then the rest of my talk I will actually just summarize, how we in Hamburg actually have been trying to respond to this very amazing crisis. Sorry, so okay, presentation mode. Right, okay. So I hope that everything technically is fine. I'm working on the assumption. So the first slide you're going to see is just about the the EMBL unit in Hamburg. So very different from Heidelberg. We are situated on Daisy campus. And things have really dramatically changed during the last 10, 15, 20 years. And I think what we have at present are actually one of the very best infrastructures for using synchrotron radiation, but also free electron laser, the European free electron laser around the world. And what is very special about Hamburg is that we are in charge of our own infrastructures. So we are running 3 beamlines for applications in structural biology, 2 for applications in protein crystallography, and 1 for small X-ray scattering. And at present, we are very much in the process of diversification. So just actually since yesterday, I should mention that we actually have started advertising 2 group leader positions, one for X-ray imaging, which will be a completely new activity in Hamburg, and the other one for electron microscopy, where the idea is to join into a new electron microscopy facility for the user community, which in this case will not be coordinated by EMBL, it will mostly be coordinated by Daisy, but we are very keen and we have been invited to participate in this new facility. So, Hamburg, like the entire EMBL, is very much, ongoingly in a transition, which is very exciting. So, what do I have to do here? Okay. So, just to give you an idea, on 2 additional slides. So, Hamburg, amazingly, is actually as old as the laboratory. So, it was founded in 1974. It was actually the Hamburg outstation originally, who actually moved into its own building, and just a year later, 1975. In the 80s and the 90s, Hamburg became really a world leading provider for structural biology synchrotron services worldwide, thanks to amazing activities of my predecessor Keith Rosen, but also other colleagues. Maybe some of you probably still remember him. And since 2012, we are now operating 3, actually less beamlines than in the past, only 3, but state of the art beamlines in structural biology, as I've already mentioned, with unique instruments including a unique instrument for time-resolved studies, where we have now opportunities really to come up with molecule movies. I don't have the time to go into further detail of this today, but it's very amazing. And at present, we are preparing for the next challenge, and this is there will be an upgrade of the so-called Petra 3 synchrotron to Petra 4 with a number of very amazing parameters. And as I've already mentioned, we are also planning to become a partner of the future cryo-electron microscopy user infrastructure. And what you see on the left are just numbers of users. I think this is from 2018, I believe. And so I think, I think we're doing a pretty good job for also, providing services to our member states. But of course, not only, also to other countries around the world, mostly European, of course. And to the last 5 to 10 years, we actually have started building up a new pillar in Hamburg, which is research focused. And at the time, we didn't know that infection biology would become a big topic at EMBL. But nevertheless, and I've been very much involved myself into this. We're doing fundraising. It took us more than 10 years for a new center, which is called the Center for Structural Systems Biology, CSSB. And we decided actually, almost 10 years ago to have a major focus on infection. And infection in different flavors, virology, bacterial pathogens, but also parasites. And we actually, so the center has opened in 2017. I myself was actually the founding director, 2013 to 2017. And this is, I think, amazing. And we have 10 research organizations actually, in this center, which is a bit crazy. If you talk to, let's say people from America, they say, you're crazy having so many research organizations under one roof. But I must say, it actually works pretty fine. So we are doing research. We are also very much going to translation. My own research focus is actually still in the field of tuberculosis, now the second largest, leading to the second largest death burden apart from the SARS-CoV-19. And we're doing a number of exciting research projects, but however, where I actually don't have the time to talk more about it today. So to the last few minutes, I just want to tell you how the Hamburg unit actually has responded to this very shocking crisis. And now now it's middle of June, and we are actually now started on looking from the kind of historical perspective. What happened to us and to all of you earlier this year, mainly in March. So just a short history is that our unit was formally closed on March the 19th, but EMBL, under the leadership of Matthias Henze, actually started very briefly asking for, dedicated to COVID-19 research and service projects, and you referred to recent meetings, from about other projects from other units. So the Hamburg unit actually was, very responsive, and 4 projects were submitted. 3 for the provision of research infrastructures by Thomas Schneider, Dimitri Serban, and Maria Garcia, all actually colleagues in Hamburg, and 1 research project. And I'm just going to show you a few slides in a moment. The research project coordinated by Christian Löw. So we started actually the Good Friday before Easter, with the first project, and that was April the 10th. And finally, our unit opened on May 11th with restricted operation. Similar to obviously many other research organizations around the world. So, sorry, I have to. Yeah. So I just give you very much an outline of, a COVID-19 project which has been initiated with Christian Löw, who is a group leader for, I think he started about 5 years ago. And he is an expert in so-called Cy-bodies. Cy-bodies are synthetic kind of nanobodies. So it's just a domain instead of having the complete antibody. And they have a technology which is called phage display, the so-called phage display technology, where they have actually started screening against basically the spike protein of the coronavirus, which is on the surface of the virus, but also on the receptor binding domain of the spike domain. And this is what you see in the upper part of the figure. And in a very short time, they found actually a number of promising Cy-bodies. Then they have characterized these Cy-bodies against either the receptor binding domain or the entire spike protein with a technology which is based on surface +1 resonance. And you see, a few figures on the lower left. I don't know the curve. I don't know. I haven't figured out how to use the cursor. Can you see it? I hope maybe you can see the arrow at least. And then, having quantified, the binding affinities in collaboration with colleagues from the Karolinska Institute, they actually measured the response to an engineered virus where the spike protein of the SARS-CoV-19 was engineered in. And very amazingly, they found for one of the synthetic bodies, of the Cy-bodies, they found 100% inhibition. And I've been talking this morning to Christian, and he claims that this is, for the moment, actually the most effective Cy-body. So, of course, other groups have been doing similar things, which actually has been identified to date. So this is quite exciting. So just going to the next slide. So what you see here is, we in Hamburg, we work very much together, and we have beamline, our beamlines, slow field, small angle scattering. So small angle scattering is very appropriate method for characterizing, let's say, in low resolution, what actually happens when, in this case, a Cy-body actually binds to its target. It is shown here with, one of the members of Dimitri Serban's group, Melissa Krebert, actually with, wearing a mask, of course, doing experiments here, beginning of May on our SAX beamline P12 in Hamburg. And so this is the result, and this is super exciting. So in a very short time, basically in a month, the group, together with colleagues from the Commercialisation Institute, they managed to getting cryogenic to microscopy structures of the RBD, the receptor binding domain of the spike protein, in the presence of, I believe, two different Psi bodies, 23.1 and 23.2. And so really providing insight what's happening at the structural level. And this is already my last slide. And the structure shows how this Psi body 23 actually binds to the receptor binding domain in an up and down state. It demonstrates that the binding site overlaps with the epitope of the ACA2 receptor, and it shows how a neighboring SB23 Psi body blocks access to ACA binding. So all this work has been done by Christian Löw, being a co-leader at EMBL Hamburg, in collaboration with Martin Helberg, who is a joint appointment between the Commercialisation Institute and the Center for Structural Systems Biology in Hamburg, and Ben Morel, who is working at the Commercialisation Institute. Now, just to finish this up, I actually this morning I've asked Christian, how do you put this into further translation? And he told me that he's made contacts to EMBL, which is our technology transfer company in Heidelberg. And at the same time, the colleagues in the Commercialisation Institute, they started actually talking to their technology transfer company. Actually, I don't know about the name. But the key thing is, of course, to follow this up, to provide this to, let's say, interested companies. Apparently, there's a very strong interest. And finally, Christian also told me that this work is now actually under review in Nature Communications, which is great. So I think with this, I forgot to mention one slide. So I would like to mention that we have Maria Garcia, who is in charge of our sample preparation characterization facility, lots of different biophysical instruments, etc. And she actually has started working together with a number of other projects which are being outlined here by making use of a range of different biophysical instruments we have at the Hamburg unit. And with this, I believe I would like to thank you for your attention, and looking forward to hear more about feedback. And we'll also look at the chat room, maybe there are some comments. Thank you very much.
Thank you very much indeed, Matthias. That was great. Certainly the floor is open if anyone has any specific questions for Matthias. I think it's very exciting to see such rapid progress also in developing reagents that can specifically recognize the virus. So do you see these having direct clinical, the Psi body applications as components of diagnostic tools, or even for treatment of patients? Could you say anything about how you see this translation taking place?
So what I forgot to mention is that the idea is actually to put the sequence of the Psi body into nanobodies and later on also into FC fragments of real antibodies. And the idea is, of course, here, I'm not really an expert on this, but I guess the idea would be to develop antibody-based drug opportunities, to block basically the infection process. Beyond all this, I have not really been personally involved into this. So, the key thing is that, there are now partners, our technology transfer partners, who are just going to pick this up and put, let's say, people coming from academic research, together with people from industry.
Okay, there's a question I see that's come up, which is, if you don't mind, I'll just read out. Are the nanobodies or any other neutralizing antibody tested against agonistic or antagonistic ACE2R activities, or is this not a risk? You can probably see the question yourself.
Well, let me just see, where is it? Okay. So yes. Certainly not tested by us, but actually we know that, this is of course done by multiple organizations and also pharma companies around the world, but is is there not a risk? Honestly speaking, I'm not really in a position, to give a very good answer to this, I must say, because I'm just not sufficiently in that field.
Okay, that's fine. One other, maybe just leading on, because this is a question perhaps that I really would give to you, but also to all the other people listening. So this sounds like a potentially, exciting development. You've got what looks like a very promising reagent. In the current situation with the global pandemic, I completely understand you were talking about, going to EMBL and looking at the tech transfer office from EMBL. It's an open question. I don't have a particular opinion about it. I'm interested in people's views. Should EMBL, if Hamburg finds a fantastically useful reagent, is the right thing to do to sort of commercialize this with a view to selling it? Or should it be to make the information or reagent available as broadly as possible so that as many people as possible can benefit from it? It's almost a rhetorical question. It's I don't particularly have a point of view. I'm just interested in your opinion, Matthias, but also the people listening, given that you're funded by the European, all the member countries.
So, I do have an opinion on this, and I think, we're talking about, openly sharing data, which we have been generating. And I think that this is even much clearer by now, also in the new leadership of EDIS, that I think this is priority number one, that, and also, the severity of the crisis, I think we just share our data. I think it's part of the idea. And then, the second priority is, of course, also looking at, intellectual property and commercialization. But I think this should really remain the second priority. This is my opinion.
Yeah, well, I agree with the comment that someone's posted that, EMBL in one way or another is funded by all the member countries. And I agree with you. I think that's a great way to approach it. Okay.
Nevertheless, we need these organizations like EMBL, to put us into contact with the right companies, because we are, of course, focused on academia, and I'm very much the exception. The majority of our people, including Christian, they're at the junior level, and they come to EMBL, to do the best possible research. So I think it's good, that we have, organizations like EMBL.
Oh, absolutely, completely. And I'd like to absolutely give a big shout out to Gabor Lam, also who runs EMBL. And I hope people don't mind me saying he was my former PhD student. I think he's doing a great job there. So maybe that's the biggest contribution then I've ever made to EMBL, was having Gabor as a PhD student, if he can help with that. No, I think this I was partly asking because I think this interface between what things are best done in blue skies academic research institutes and what things are best done by organizations that have the expertise and focus to actually deliver things. We're going to come, I think we'll come back to this point. It's a very interesting point when Olivia gets to talk, who's also working in the US in a government-funded organization. But, it's one thing to discover something that could be a potentially clinically valuable vaccine or therapeutic, making billions of doses of it and finding a way to deliver it to all the people who need it around the world is something that I don't think any basic research institutions are really designed to do. And so finding that way to have the interface, I think, is a really important thing that, again, I'm hoping the Alumni Association can play a significant role in, as we have many alumni who've made this transition from basic research to working in companies. But Matthias, that was great. Thank you very much. I'd like to move our continents now and zoom across to the US. And we have the next two speakers are both alumni from the US. So if I could introduce, first of all, Patricia Kam. Hello, Patricia. Thanks for joining us. Perhaps you could just remind people about your own history at EMBL.
Okay. Yeah. Hi, everybody. Very nice to be here. It's funny. My screen is telling me that I am muted, but I guess I'm not.
No.
Okay. Yeah, I am from an earlier era at EMBL, so from back in the 80s. I see a few familiar faces there, Angus being one of them. And that's back when I was still a wet scientist. And I started out in the differentiation program and then dried out by going through bioinformatics when that was still based at Heidelberg in the pre-EBI days. And over time, I have drifted farther and farther away from the lab, and now have been working for the last couple of decades, actually, in global health, and in a different area, in a research, writing, and editorial type role. For the last decade, I've been at Doctors Without Borders at the New York office in the U.S. So this talk will be a kind of a change of gear from what you just heard. And what I'd like to do is tell you a little bit about how Doctors Without Borders is responding to the outbreak. Let me share my screen. Before I get into COVID, I just want to give you a 1 or 2 minute introduction to Doctors Without Borders, which also called Médecins Sans Frontières, and I'll refer to it as MSF in this talk. And I'd like to tell you just give you a quick overview of who we are and what we do. Although our name is very well known, people often have only vague ideas or sometimes wrong ideas about us. So you may know we're a medical humanitarian, a nonprofit organization, and actually a relatively big one. We are working, this is a pre-COVID map. So pre-COVID, we worked in about 70 countries around the world, delivering medical care to people based solely on need and not on political agendas or political sides in any way. Neutrality is a very core part of what we do. And in these countries, the focus is not on duplicating services, but focusing on areas where the countries themselves where there are gaps, either in countries that have fragile or weak health systems, where there are populations that are excluded from care. This is a 2018 map. As I said, at that point, we had about 450 projects in these 70 countries around the world. And field staff of about 43,000. And another kind of myth about MSF, which I'll now bust, is that of those 43,000, the vast majority, something like 90 or 95%, depending on the project, are actually national staff who come from the communities where we work. So MSF is thought of often from afar anyway as an organization that has expats flying in to deliver care and to do the work, but it is really overwhelmingly local communities who we are supporting and often guiding in doing this work. This slide gives you an example of the types of activities we do. And I want to mention this because also it's part of what's frequently misunderstood about MSF, and because it's also very important in terms of how we're responding to COVID-19. Lots of people know MSF through the responses to crises that make the headlines, earthquakes and famines and wars and other disasters like this. And this is an important part of what we do, but actually a relatively small part. A very high volume of the work we do is around epidemics, and you can see, let me see, some of that here. I've just listed meningitis, but cholera, Ebola, measles, we're now fighting one of the largest outbreaks, certainly the largest outbreak on the planet and one of the largest in many decades in Congo and Chad. And other outbreaks that are just always popping up in one place or another. We're very active in treating malaria and in preventing malaria as well. You can see here 2 million consultations or 2 million treatments. In 2018, we run hospitals that do surgeries. We do a lot of mother-child care in places that have very high maternal mortality. We're active on HIV and TB. And in those two diseases, particularly in areas that are difficult for countries to manage. So we manage, often the harder cases, the drug-resistant cases, for example, or people who are severely ill. And those are also, HIV and TB are very active areas of our research program as well. So now let's turn to COVID. And here is a map which is already quite out of date. It's about a month old. Just lost one ear here. Hold on a second. Yeah, this is where our COVID response stood about a month ago. I don't have a more recent map than this. And the black dots show the places where we had specifically COVID-19 dedicated projects. And it illustrates two of the many challenges we face, and which I'll talk about in the next slide. Number one, you'll see dots in countries that were not highlighted on my earlier map. So, for example, the United States is not a place where you typically think of Doctors Without Borders working, or you'll see most of Europe has dots. And I'll tell you in a moment about what we're doing, which is quite limited in these countries. But that was certainly new for us. I think the other incredibly huge challenge has been that this crisis has unfolded essentially around the world. In contrast to past crises and pandemics, bad as they may be, they are almost invariably localized. So there can be a brutal war going on here or an outbreak going on there, but everything else is essentially carrying on as normal. COVID is unlike that. This is a planetary crisis, and really all around the world, people are either countries, communities are battling outbreaks, or they're preparing to battle outbreaks and to stay safe and to take measures that keep COVID at bay. And I don't think MSF has ever really had to gear up simultaneously in all across the world like this. And of course, the scenarios in these countries are not the same. There is no one-size-fits-all kind of solution. So it has been just a huge scramble to respond. And for many months, this has just completely dominated the work of the organization. So now let me show you a little bit about, let me build on that notion of the different challenges we're facing in responding. I mean, first of all, as I said, and I didn't list this, but it's in a context for what I'm saying here, is that while MSF is a big organization in the scale of medical humanitarian organizations, we are minuscule in the face of a crisis like this. So we are a tiny player with very little capacity, I would say. I mean, we have a lot of capacity as an organization, but relative to what's needed, obviously, we are small. Like everyone trying to respond to this, we're faced with uncertainty, with tremendous uncertainty about so many things around COVID. We know very little about where it is outside of the European and outside of the high-income countries, I would say. So when we look at the parts of the world where there are a lot of fears about rampant spread and even more limited capacity to respond, looking at Africa, looking at the poor parts of Asia, and so on, we know very little about where it actually is. Some of the numbers across Africa are really relatively low, at least compared to the worst fears. Does that mean there are not many cases? That COVID is just taking its time getting there and may spike or peak? Or does it mean that they've been largely spared because many of the countries did lock down early? Does it mean that there are huge numbers of asymptomatic infections? That's perfectly plausible, considering that the population on average is much younger in Africa. or are we just not seeing the cases and the deaths because they're happening at home and out of sight and in remote areas and so on? So, and we certainly are seeing some places. I give Yemen as an example, where Yemen is a country that's facing a horrible humanitarian crisis already because of the wars. And now we're seeing sudden spikes in extremely severe cases, people appearing at care very, very late and dying extremely quickly, and younger people than we're seeing elsewhere, largely, young men in their 40s and 50s. Not supposed to happen. As I mentioned, there's very little testing able to happen in many parts of the world. So again, we have really little insight into what's going on, and to a large extent, we are flying blind. One of the huge problems we faced, as have others, this is not specific to us, is with supply chains for the equipment we need, the protective equipment, and for basic drugs, antibiotics and malaria medications and things we need to keep our programs running. There's also been a real surge in poor quality goods flooding the markets, bogus masks and bad PPE and counterfeit drugs. Those are always issues, but they have really surged during this outbreak, and are the amazing, absolutely incredible logistics people we have working for MSF, and who are the sort of unseen beating heart of the organization, have done amazing work on quality assurance and on getting supply chains back to a stage where we can now say a few months in that we are already in better shape. Nobody is out of the woods, but things are getting better. The border shutdowns, difficulty transporting across country lines, transport being blocked and so on, have been in just huge obstacles. And the same obstacles have applied to moving people. And this has had a huge impact on our project, because even though our programs are very largely community-based health workers, we do still have to move a lot of people around, and that has come to almost a screeching halt. And that's been as much because of the external situation as, at least at the beginning, caution on our part of not wanting our staff to be ones introducing COVID into communities where it did not exist. So here I will go through 3 sort of top-level priorities in terms of how we're responding. And again, the response has been the volume and the range of activities across all of these different contexts has been vast. But I'll try to, I boiled it down here to a few key things and a few examples. So one of the first things we did was, and this was when COVID was really emerging in more high-income settings, so starting when it left China into Hong Kong and then coming to Europe and the US. This was we began projects to focus on people who were left out of the sort of the national responses and the more formal healthcare systems. So we, as an organization, very experienced in helping to, for example, do infection prevention and control in settings where we're in a situation where many people cannot isolate at home and wash their hands all the time because they don't have a home, or because they live in an overcrowded refugee center, or they're prisoners, or they live in they're undocumented migrant workers. So these are the populations we focused on in the United States and in Europe. And we did a lot of training of staff who run these kinds of facilities, a lot of direct work in communities in the US, for example. There's a big article today in The Guardian about Immokalee, Florida, where there's a large community of farm workers, undocumented farm workers who harvest the food that people who are safely staying at home are depending on, but who themselves have very little access to care. So we've done a lot of projects in helping to find ways to keep those people safe in the very constrained situations they live in. And these are some of the tools we use: direct education and health within the community, where there are living facilities like homeless shelters, working to train the staff, provision of shower facilities, hand washing stations, soap and help learning and clean water sources and things like that. I've lost one here. There we are. This priority is, this was our parallel priority, I would say, and that is to maintain the essential services and the emergency responses that we are already doing. And all of those things I showed you in an earlier slide, the TB programs, the maternal, the safe birthing facilities we run, the malaria services. Malaria season is upon us. I told you about this major outbreak of measles in Congo, vaccination campaigns, as the developing countries have instituted shutdowns as well, and there were shutdowns quite widely across most of Africa, for example. A lot of these programs had to be suspended, and we have worked hard to, so here are a few examples of the kinds of programs we've had to postpone, but that we're working now to get back on their feet, and many of them we already have as PPE has become more available and as we have found other ways to adapt how we work. Keeping our clinics safe has involved setting up proper triage at the facilities, of course, but also moving out of the clinic to wherever possible, doing more mobile clinics, for example, and adapting our programs so we can do more and more things virtually in ways that do not require face-to-face contact. And actually, even in many of the poor settings where we work, people do have cell phones. So we have been able to do quite a lot of work digitally. And the third part of our response is direct care for the patients themselves and for affected communities. And this work has involved everything from surveillance and contact tracing in communities to repurposing some of the hospital and clinic facilities we have to directly care for patients. We have not been able, these are for the most part completely not places where ventilation, for example, is available, but a big challenge, but one that we're doing decently and being able to address is finding ways of at least being able to provide oxygen in these settings. The supply of oxygen has really been difficult, but in any case, we are able to set up some hospital facilities, but also isolation facilities in communities for people who are infected but don't require hospitalization but can't isolate at home, and various other kinds of direct care that we can offer. And my last slide here, since you are a group of researchers, is just, and I'm sorry, I do have to speed up here, is to talk about skim over the kinds of research we're doing to try to improve our response. We're doing a lot of operational research on how to adapt and innovate these programs. I'll skip the examples for now, but just to be effective in these kinds of circumstances and make sure that the changes we're making are still maintaining some quality of care. We're looking at seroprevalence in some of the communities where we work to try to get out of having to fly blind. Another important area, which is still under discussion, but a lot of the there are many, many clinical studies going on around the world for treatments. We are specifically focused on those, not that work sort of at the end of the disease progression, on people or late in the disease progression on hospitalized patients, but much, much earlier, and looking for existing drugs, ideally, that can prevent progression from mild disease to cases that do require hospitalization. And I will leave it at that, and thank you for your attention.
Patricia, thank you so much for sharing that experience and just for working with an organization that is willing to take on just impossible problems and make such a big contribution to them. I'm sure there's lots of questions, and I'd really like to explain. Just if I could ask one thing, however, right at the beginning, you said that Medicines Sans Frontières is a non-political organization. And my obvious response to it is, how can you be a non-political organization in a situation that is so inherently political and is manipulated by so many people? America is accusing China of creating the virus in its labs. China is accusing America of doing something similar. Brazil's pretending there isn't a problem, and it's all a manipulating. Goodness knows what North Korea or some other places. Apologies if anyone here is from North Korea, but healthcare is such an inherently political issue. I mean, maybe if you could just share with them, what was it like to be in this organization in New York? Because that must have been one of the most politicized centers in America in dealing with this epidemic.
Yeah, well, and that is true. All of that is true. And that is really a great question, and one that we talk about a lot and we think about deeply and carefully in the way that we are public facing. I mean, we are also an organization that lives entirely from donations from private people. 95% at least, 95% of our funding is for private individuals. So maintaining this neutrality in a public sense is important, both for credibility with our donors, but it's also essential for getting access to patients in the countries where we work. Once we're seen as a political actor, that becomes harmful. So, of course, being in the United States at this time is difficult. Looking at the way my own country has politicized this, and the best answer I can give you is that we stay very focused on the medical needs and on the medical data we're seeing. We are focused on helping people in need. We see where the need is. We assess it. We look at what our evidence tells us about what works. We are very experienced responding to outbreaks, and we know what needs to be done. And to some extent, we try, the rest is noise. Sometimes we have to confront the noise directly. And Angus, we were chatting about this a little bit before the meeting started. In the US, for example, the policy at the southern border has certainly been very harmful to the public health scenario we're all facing, where undocumented people who got across the southern border and were detained in extremely overcrowded and unhygienic settings where COVID was absolutely spreading were then deported, just dumped over the border back into Mexico, where they certainly contributed to bringing more cases back into that country. We have been very outspoken about that. But again, so you could say that is political, but we're speaking about the data. What we see is that this policy leads to harm, it spreads disease, and therefore we speak out against it. And sometimes to be neutral does require a certain amount of being political. So that is the strategy.
That's the catch-22.
Yeah.
So, that was fantastic. I hope you can stay a little bit longer, and I encourage people to come back with questions. I'd like to move on to Olivia and introduce Olivia Steele-Mortimer, who was also at the EMBL in Heidelberg, who could perhaps just say a little bit about your background at EMBL. And then I think this comes in beautifully to the point of working for a government-funded research organization focusing on human health. Olivia.
Yes, thank you. And hello to everybody from Montana. So I work for the NIH. I was at EMBL 90 to 94 in John Grunberg's lab as a pre-doc, and did basic cell biology with the aim from when I started at EMBL that I wanted to go on and work on intracellular bacteria, which at the time, there were not many tools available. And I was just really lucky to be at the right time, at the right place, the right time. And from there, I went to work in Vancouver, British Columbia, with Brett Finlay on Salmonella. So I've been working on Salmonella since 1995. And I was hired here at Rocky Mountain Labs 20 years ago. I have my own lab. And I work on Salmonella, not on viruses, but I'm going to talk to you a little bit today about a project that we're doing to develop a vaccine using Salmonella. And I do have just a couple of slides I want to show, so I will try and share my screen here. So just a brief introduction to Rocky Mountain Labs. We're actually part of the NIH intramural program. So I'm sure you're all familiar with the NIH main campus in Bethesda. Out in Montana, we have this very small institute. It's about 450 to 500 people, and 3 labs, and they're all working on infectious disease of one kind or another. So the lab that I'm in, its equivalent to a department, is the Laboratory of Bacteriology. And we have a very long history of working on tick-borne pathogens. So Borrelia burgdorferi is named after Willy Burgdorfer, who worked here and was the first person to image Borrelia on an electron microscope. And we have the Laboratory of Persistent Viral Diseases. This is headed by Bruce Cheeseborough. Many of you will know for his seminal work on prions. We still have amazing prion research going on here. And finally, we have the Laboratory of Virology, which came here, was opened in 2008, is headed by Heinz Feldman, who got his PhD from Marburg. And Heinz heads a group that is really looking at all of these high containment, what we call high containment viruses: Ebola, Marburg, high pathogenicity, influenza, SARS, MERS, and of course now COVID. We have a high containment lab with BSL-3 and BSL-4 facilities. We have a non-human primate facilities. So a lot of the animal modeling for COVID-19 is actually going on here in Montana. And right now, our guys are collaborating, for example, with the group in Oxford. Everybody's hearing about the Chad vaccine from Oxford, is this chimpanzee adenovirus. The animal testing for that virus was done here. And I'm happy to answer questions about that later. They're also doing testing for VSV vaccine platforms, RNA, different forms of deliveries. They're doing testing therapeutics. So, for example, they tested remdesivir in infected non-human primates and simian macaques, and it showed that if you treat with remdesivir starting very early in infection, you can get improvement. And they're also, right now, for example, they're testing hydroxychloroquine in animal models here. They're using, right now, the main models they're using are hamsters, because the ACE2 receptor in hamsters works for SARS-CoV-2, obviously non-human primates. And they are working on mice, but these are the humanized mice with the human ACE2. And we're actually collaborating with another old EMBL alumni, Nadia Rosenthal at Jack's Labs, to get better mouse models in the long term. So those are some of the things going on. Clearly, since the beginning of February, our small institute has been absolutely focused on this pandemic and doing that basic research, but also doing testing of therapeutics and vaccines. Since March 20th or 16th, we've been essentially doing only COVID research. So those of us that work on other minor diseases have all been sent home. But the BSL-4 labs have been working 24/7. It's absolutely incredible. So to transition to what I've been doing, so my lab, although I work on Salmonella, I have a project in my lab trying to develop a therapeutic vaccine for breast cancer. And so there's a lot of interest in using Salmonella and other gram-negative bacterial pathogens as tools to either vaccinate for or treat cancers, and particularly solid tumors. And I don't have time to go into it today, but basically what we're trying to do, we're collaborating with an amazing breast cancer researcher at the Huntsman in Utah, and she studies metastases. So the idea is we're developing a vaccine that would treat you would give personalized medicine. So you would give for a specific cancer that somebody has after they've had surgery in order to prevent metastases of the disease. So what we had done was we had developed a vaccine platform. We're still at the very early stages of this, but we had been discussing with the virology group here if we could use this platform as a sort of proof of principle for one of the viruses that they're interested in and studying. And so the one that we finally settled on was not a coronavirus, but was actually Lassa virus, which is endemic in parts of sub-Saharan Africa. And rather than go immediately into human modeling, we have a great system, animal model here, using the natural host for Lassa, which is a Mastomys natalensis, a rodent that's not quite a mouse or a rat, somewhere in between. And that's the natural host in Africa for Lassa. And so our idea was if we could vaccinate that host, we could prevent Lassa getting into the villages. So we could just put Salmonella is very easy. It's very resistant to desiccation. You can sort of put it out in the food and just have these rodents help themselves and ring vaccinate effectively villages. So we're working on that when this pandemic started. And so the virus lab asked us if we would join with them and try and see if we can make a Salmonella platform vaccine for COVID. And so I will go to the next slide. And basically what we're trying to use is a live Salmonella. And importantly, it's both attenuated and self-destructing. Because obviously it's not enough that it's attenuated, but you need to get delivery of the antigen, or in this case, DNA. We're using it as a delivery system for a DNA vaccine. So we want delivery of a plasmid into cells, which can then express the viral antigen and hopefully mount a response. So why would you choose to make or use Salmonella as a vaccine? It's been studied as a possible vaccine platform for well over 10 years. And some of the reasons are, really important one is that it's extremely simple and inexpensive to manufacture. So basically, you just have to grow the bacteria in fermentation. If you can consider, on the other hand, actually trying to purify enough nucleic acid to do either RNA or DNA vaccinations without a delivery system like this, I've heard people talking about having to make maybe 80 kilograms of RNA to vaccinate large numbers of people. So, I mean, the logistics of this is just so different from purifying RNA. Salmonella are remarkably stable. They will survive for long periods of time at room temperature, on different surfaces, etc. With an oral vaccine, clearly you don't need any needles, no medical expertise is required for administration, and especially in the third world, this would be a huge benefit. And it can actually induce both mucosal and systemic immune responses. This has been very clearly demonstrated in mice, in mouse models. It has not so far been so clearly demonstrated in humans, and I'll come back to that later. The other thing is that in Salmonella, you can potentially make multivalent vaccines. So there's enough space to put multiple genes. So for example, you could maybe make a vaccine that would vaccinate against Ebola and Marburg and Lassa all in one platform. So just as a quick introduction to how Salmonella infects us and how we can use this as a vaccine delivery system. So the things that I'm interested in, coming from a cell biology background, is really how Salmonella interacts with specific cells in the host. And the ones that we look at are really intestinal epithelial cells and macrophages, and primarily intestinal epithelial cells. And so Salmonella is ingested orally, and it survives at least well enough through the stomach, through the acidic environment in the stomach to get into the intestine. And there, what's very important is that it interacts with mucosal cells, in particular epithelial cells, M cells, and can be picked up by dendritic cells. And so work in my lab has really focused on what's happening in the epithelial cells. And I don't know if you can see my cursor, but what you should be able to see is that bacteria actively invade. So they have a very sophisticated system to inject bacterial proteins across host cell membranes and induce their own uptake. And once they're internalized, most of them replicate inside of a vacuole, a modified phagosome. And this is where most Salmonella research and really focused on intracellular replication. However, on the right here, the cell with blue bacteria in it, my lab has shown, and several other labs now too, that a certain percentage of Salmonella escape from the vacuole, and they survive and replicate in the cytosol of the epithelial cells. And they don't do this in all cell types. They can't survive and replicate in the cytosol of macrophages, for example. But we've got a paper right now that's going through review where we've shown that this replication in the cytosol of epithelial cells is really important for persistence of Salmonella and spreading. So animals that have Salmonella can be infected for years, and in mouse models and also a calf ileal loop model, we've shown that this particular population in the cytosol of epithelial cells seems to be really important for maintaining that infection. So one of the issues with Salmonella vaccines in humans is that they seem to persist more than you would like. So you really don't want people that are vaccinated to be shedding the vaccine strain for weeks or months after vaccination. And so our recent findings that these cytosolic bacteria are really important for shedding has given us something to target to try and limit that. And so what we've actually done now is we've introduced a phage lytic gene or multiple genes under a promoter in the Salmonella that is induced by glucose 6-phosphate. And glucose 6-phosphate is very highly concentrated in the cytosol of epithelial cells, other eukaryotic cells. And it seems that the only place where we see expression of this lytic gene is in the cytosolic population. So we can effectively block replication in the cytosol, and that blocks shedding over time. So we've only done this in mice, but we're pretty confident that this would also help in humans, or if we use this vaccine platform in other animals. And not only does it stop replication, but it actually lyses the bacteria. So if you have a DNA plasmid in those bacteria, it will release it effectively, and it can be transported into the nucleus of the epithelial cell, and you'll get expression of the viral protein. So in this case, our vaccine is expressing the spike glycoprotein, which is what most of the vaccines are going for. And not only that, we're not expecting all of the plasmid to be released or to interact only with epithelial cells. You have other cells that are like macrophages and dendritic cells that are constantly interacting with the cells in this mucosal surface. And so we expect that the plasmid will ultimately be taken up by antigen-presenting cells, such as dendritic cells in the gut. So we have several mutations in the strain. Just to give you a sort of brief sort of overview of what kind of attenuations we can do for these Salmonella vaccines. First of all, I don't want to forget that so far there is one live Salmonella oral vaccine available. It's been available for a long time. That's the one against Salmonella typhi or typhoid fever. And the strain is called Ty21A, and it was actually made before we had really sophisticated molecular biology mechanisms to target specific genes. It was chemical mutagenesis, and so it has multiple mutations in it that attenuated. But this is just a good example of something that it's a very robust, still a very useful vaccine. For Salmonella typhimurium, which is the strain that we use that causes food poisoning, not systemic disease, unless you have HIV, AIDS, or other immunodeficiencies, there are quite a lot of mutations that have been studied in animal models, mostly mouse models. And so if you, for example, knock out aromatic amino acid biosynthesis, you can get mutants which cannot replicate well in a mouse model, but they can elicit very robust immune responses and protect animals against challenges 10,000 times higher than the LD50. There's also...
Olivia, we're running very close to running out of time.
Okay.
So I didn't want to interrupt you, but if you could perhaps, yeah.
Yeah, but this is basically it here.
Okay.
So our strain has basically got some mutations in cell wall biosynthesis, and we have another strain here with the aromatic. And so what we still don't, in mice, we know that these basically have been shown to work previously for influenza, for example, but we have to try it in hamsters for the COVID because that's the only really robust small animal model we have right now. And in humans, it still remains to be seen whether or not this will work. We're hoping that our additional lysis, so self-destruction, will make it safe or safer in humans, but that's down the road. And even if it doesn't work in humans, potentially this could be very useful for vaccinating animals. So, for example, now we've seen that cats, big cats, domestic cats, civets, other animals are susceptible to these diseases. So it could be useful for that. And I'll stop there. Thank you.
Okay, thank you very much, Olivia. And in fact, if we just come back to the question, I'd like to open up to anyone who'd like to comment or speak on any of the topics raised today, or indeed on topics from the last four meetings. But Olivia, if we could just come back to this point, you're working in an institute centrally funded, I believe, by the US government. Is that correct?
Yeah.
So if you find that your technology, it is a fantastic route to treating the current pandemic or a future pandemic, how does that work in terms of sharing the technology? Or do you have a tech transfer office? Do you try to set up a company? Would the NIH say, okay, there's no time for that. We want everyone in the world to have a vaccine that will cure them of COVID? Or does the US government try to say, now we have an economic lever to gain an advantage over our I'm sure you don't perhaps have all the answers, but I'm just interested to know if as a researcher in the institute, that's something you discuss or have input into.
Yeah, well, we do actually have a vaccine research center within that NIAID on the main campus in Bethesda, and so they've got a real history of developing new vaccines, and they have different agreements with various companies. So the goal is that any of these vaccines should be made available as soon as possible, and it's not about the American people benefiting, at least from the NIH, from our leadership. Clearly, we have been hearing other things, that anything that's available should be kept for the American people, but that's not from within the NIH. We're certainly aiming to have things available. Whichever way we can do it, in collaboration with companies that we have agreements set up with. But this is something that we may have to get completely new because it's just so different from all the other vaccine projects here.
Yeah, I think it's very exciting developments. And maybe actually we have in past meetings also had a chance to ask questions to the audience and get some feedback. Again, in the participants tab at the bottom of your screen, if you click on that, and you can see everyone who's taking part, and you can also see the green and red yes and no buttons. So if I could just ask a question, do you think if researchers are working in government taxpayer-funded research institutes, that the outputs of their research should be simply provided freely or commercialized through some other route? So do you think organizations such as EMBL and NIH, that all the commercial research or potentially commercializable research should just be publicly available to the community? Yes or no? So I don't know if people can have access to seeing that. Nusha, Peter, perhaps you could tell people. At the moment, actually this is the most unanimous output we've had for or response to any question I think we've posed. At the moment, we're on 100%. A lot of people replying. We're seeing 100% response so far.
I think this is Peter adding the same question online so that you can do it again and then you can see the answer.
Okay, I'm seeing something different on the polls, but anyway, I can see on my screen, if it's not visible to everyone, it's almost, is it that 28 people have said yes, it should be publicly available, and nobody has said no, it shouldn't. So that's about as conclusive a result as you get. That's very interesting, and also I guess something we can feed back. Are there any more questions at the moment, or does anyone like to comment on this or any of the other issues that have been raised?
I have a question. If it's possible for you to hear me?
I can hear, but I can't see who I'm.
It's me.
Yes. Michelle, yes, please.
My question is, if this potential development is not patented by a government institution, is it then liable for patenting by somebody else? So is it then necessary to protect it from exploitation from different commercial interest by patenting it under the auspices?
It probably would be patented by the government, but in a way that it can still be, shared and available. They are pretty careful about patenting things these days.
Thanks. That was my question.
Thank you very much indeed. I'd also like Uli. So I see a question he's written. I'd like him just to be able to give his question, because he's saying I asked the wrong question, but I want to argue with him about it. So please come and is Uli there? You could. Hi. Do you want to share your?
Okay, as a patent attorney, I would say passing information away is always good, but you always have to think that having produced an antibody or whatever at a stage to have it commercialized, then there is a huge gap thereafter, which needs, you have to bring it into the market. I mean, you have to convince people that it's needed, that there is no better other product on the market. I mean, there is an enormous commercial risk. I mean, whether would mean the antibody is as good as proposed, would mean, or whether would mean it's in conflict or produces, and let's say would mean immune storm. Would mean this needs all to be tested, and you need, in general, would mean a huge amount of money. Would mean to have it, let's say, to convert an invented product into a commercial ready product. Would mean commercially ready product. Usually would mean I see it from my clients, from my pharmaceutical company, from my diagnostic company, that such takes usually 7 to 8 years, would mean from, let's say, development, seemingly ready, until would mean market entry. And these 8 years need to be financially covered. And for this, you need an IP.
Yeah.
So it would mean a patent is needed to bring something into the market and not to, let's say, keep others out. That's as simple as that. Would mean
So I agree with you about that, actually, really. And I think to be fair, there are some differences. It's just a discussion point. I think it's not a simple question. I think one difference, if you are talking about clients who are in biotech or pharma companies where investors have put money in, and absolutely they need to protect their intellectual property. I guess one of the things that people are just discussing is if we as taxpayers fund research, to what extent should that output of the taxpayer-funded research be made available also to your clients and pharma companies so that they are given more freedom to try to develop products? But I think it is a very complicated process. And I see, Francis, you've got your hand up. Do you want to comment on this?
Thank you, Angus. So there's a weird thing here with Zoom, because I voted a no. You had 28 votes yes, but I pressed no, and then I realized that when you press the raised hand to say something, then somehow the vote doesn't work. But whatever.
Okay, it was still overwhelming. Even if I asked the wrong question, and even if you reversed it, it was still overwhelmingly people voted yes.
So I think absolutely researchers who are operating on public money from the taxpayer or from charitable donations or whatever need to engage in protecting the intellectual property because they need to retain control of that intellectual property. If they do not keep a foothold in control, it will be exploited by someone else. And that someone else is just going to try and make a profit out of it. Whereas we as, publicly funded researchers need to stay in the conversation. And ideally, to keep a veto on the commercial exploitation so that in the cases of extreme public interest, we can say, this is not about making a profit.
Yeah, I think I broadly agree with you, Francis. I should just, before I'm branded a hypocrite, I should declare I actually have launched a spin-out company, and I have some IP in certain areas.
So have I. And I also share your comments about Gabor and Emblem. He's done a great job and all of that sort of stuff. But from my experiences, I really believe this is a terribly important point, because if we are just sort of idealistic and naive, and we just release our information into the public domain, believing that it's going to be for the good of mankind, I can assure you it will not be. It will be exploited by people who are only interested in the pursuit of making money for themselves.
So I think that there is a lot. If I could just share also my own comments, but I'd love to hear again from everyone else's. I think there's a lot of naivety in the academic community about what IP is and how it translates into products. I mean, most patents don't make money, but I think it is important that there's a transparent process available and more understanding of how to properly protect IP so that it can be made more available in a controlled way. I have personally, my biggest ethical problem is when some companies or people get hold of patents and use it to prevent people doing things. That's where I think it can be misused personally. So I think actually one reason for institutions such as EMBL or NIH protecting their intellectual property is actually to ensure that people do get access. And if they pay a license, that's fine, but that they get access to properly use it and don't end up preventing people using it. And in a crisis like the COVID epidemic is probably something that highlights a real ethical challenge where it would just seem wrong to me that one small or even large company could suddenly hold the rest of the world to ransom and essentially decide who lives or dies based on what they do. I think that's not ethically acceptable to most of us. At least I hope not. Any other questions from the community at this stage? Anyone else with a hand up who'd like to contribute or speak? I do think this topic is a really important one for EMBL as an organization and for all of us, actually, as alumni who can contribute in different ways. Bertold, you have your hand up. Would you like to comment, please?
Yes, just a short comment. Since I'm working at the European Patent Office as a patent judge, actually, the scenario that you just pictured, that patents could be used to then prevent people getting medicine, essential medicine, like in a crisis case like we have today, I think is not very likely, because most jurisdictions have actually legislation in place where in such case you can force a patent holder to let others also produce the patented invention. And that has been done in the past in several jurisdictions. I know of the US, of Brazil, even Germany has this kind of jurisdiction. So I think what Uli mentioned is very important, that it's not only about getting the information about a new medicament, but it's also about getting the funding for bringing that to the market and going to clinical trials and so on. And that in many cases needs a lot of money and needs investors, and that needs securities. And often the IP is the security. So I also agree with Francis that having the IP puts you in control, and then you can get into negotiations with companies and with investors and so on. But it's a very important topic, and there has been a lot of discussion also in IP circles now in these times about what's the best way. But I see quite some willingness also on the side of pharmaceutical companies to engage in collaboration and to make a big effort to bring medicaments and vaccines as fast as possible to as many people as possible.
So thank you. That was an incredibly helpful contribution, and I definitely appreciate it. I wasn't aware of this special mechanism in extreme situations for preventing someone holding up progress. I have seen some examples in perhaps less extreme situations. I fully support the idea of, like Francis. I think Francis and I are largely saying the same thing, maybe in slightly different ways. I think it's an incredibly important topic for how science and scientists can deliver impact of real benefit to society. And the real way for us as researchers to pay back the taxpayer and society for supporting us is to deliver impact in many different ways. And that's by finding out important new things, like Olivia's, pioneering some basic research into new ways of creating vaccines or treating disease, or bringing inventions to the market. And I think part of that whole business is the tricky part of translating from basic research into commercial applications. And all I would like to say is I think it's one that we haven't, as a community, got perfectly correct yet. And that there's an opportunity to improve it. And I actually think the Alumni Association and the breadth of expertise and capabilities of our alumni could actually make a significant positive contribution to this. I think it's a topic that maybe we could raise again in the future. I think that we've come to the end of today's session. I can't believe how fast the time has gone past again.